Hey everyone, we're going to kick off our energy unit with a discussion on enzymes. When we talked about macromolecules, we talked about one of the roles of proteins is to be enzymes. So enzymes are going to be catalysts in that they're going to help a reaction to occur. They're going to lower the amount of energy that reaction would require for it to get going. So we can think about reactions as having an energy barrier between the reactants and the products. So here's our energy barrier. Right? We have reactants coming into the reaction, and then we have products coming out. Right? So this would look like reactants. Reaction happens, and then we get products. Now the way you can think about this is that in a reaction, you're either trying to build something or you're trying to break something apart. So molecules don't just do this spontaneously unless we have some kind of input of energy. So if you think about like being linked arms with someone else, right, you're going to stay in that linked arm position until you start moving. And you're going to be more likely to be unlinked if you're moving quite a bit. And that movement could come from being heated up. So if you heat up, the molecules move faster, so they're more likely to break apart. If you're trying to build something together, then these things that you're trying to build would be like bumping around the room, and if the temperature is low, they'd be moving around slowly and be less likely to bump into each other and bond. If you heat them up, then the molecules or things are going to start moving around a lot faster, and if they're moving around a lot faster, they're more likely to bump into each other, and then a bond could occur. So the easiest way to get a reaction to occur is to heat something up. Heating it up is going to make the reaction speed up or happen more quickly. But living systems can't always heat up when they need to speed up all their reactions, so they need some help. If we look at this bit over here, right, if these are our reactants here, right, the reactants are in a stable state, and then when they get heated up, right, when that energy gets input, they're in a less stable state, and then they will tend to move through the reaction. So the energy that we need to input into a reaction, right here, right, that's called the activation energy. So activation energy is the amount of energy we have to input into the reactants to get a reaction to occur, to rise over that energy barrier. So we can see over here, right, the blue line is going to be the reaction without an enzyme, and then the red line is going to be the reaction with an enzyme. So what the enzyme is doing is it's going to lower the activation energy that is required for the reaction, and if the activation energy is lowered, the reaction can happen at a lower temperature and it can happen much more quickly. So let's talk about this free energy thing over here, right? This would be Gibbs free energy, which is written as this, right? And we can think about Gibbs free energy as the available energy in a reaction. And another way to think about this is that bonds store energy. So when you create bonds, you're going to store up energy in those bonds. So you're going to have a greater amount of available energy. If you're breaking those bonds, you're releasing that energy. So you're going to have less available energy. Right, so we have in these reactants here, we've got stored up energy in the bonds. The reaction happens, we have that activation energy input, reaction occurs, and we get these products. And you can see here that the products have less available energy. So we released energy, right? Since the products have a lower amount of free energy, we had a drop in free energy. Since the products have a lower amount of free energy, we released energy during this reaction, which means we likely were breaking some bonds. So we're going to think about reactions as being either endergonic or exergonic. And that endergonic versus exergonic distinction is going to depend on if there is a drop in Gibbs free energy, if there is an increase in Gibbs free energy. So remember that bonds store energy. So if we are making bonds, then we are storing up energy, so we should have a greater amount of available energy. If we are breaking a bond, we are releasing energy, so we should have less available energy. So if we think about that graph again, right, we have that available energy, Gibbs free energy on the y-axis. If that change in energy is negative, which means it's going down, right, do we think that's going to be an exergonic reaction or an endergonic reaction? Exergonic means that energy is exiting the system. So think exergonic, exiting. Right, so I'll give you a second to answer that. right? It's going to be an exergonic reaction, right? Because energy is exiting the system, so we have less available energy. If we have a positive change in G, right, so that the available energy is going up, we're going to have the opposite happening. So a positive change in G gives free energy is going to be endergonic, where we're storing up energy in bonds. So this is what the reaction graphs look like for those, right? So an exergonic reaction, we can see that the products are less than the reactants in terms of available free energy. So we have a negative change in G. Right? And we can see that endergonic reactions, the reactants are lower in available energy than the products. So we're storing up energy, so we have a positive change in G. The exergonic reactions tend to be spontaneous, right? These will be more likely to happen spontaneously, whereas the endergonic reactions are going to be non-spontaneous. Both of these reactions can be helped by an enzyme, and in both cases, the enzyme is going to lower that initial activation energy. So both of them can have a change in activation energy, right, by an enzyme, so therefore happen at a lower temperature and happen more quickly. It's going to be important to remember exergonic versus endergonic, and then also remember which graph would go with which kind of reaction, and if that means that energy is being stored up in bonds or if energy is being released from bonds. Another way to think about reactions, another naming scheme for reactions, is if they are catabolic or anabolic. So this has to do with whether or not the reaction is breaking apart a molecule or if it's building up a molecule. So in our catabolic reaction right here, we can see that this bonded substrate comes in to an enzyme, and then it gets broken apart into these two distinct pieces. Right? So in a catabolic reaction, we are going to be breaking bonds. I often think like catabolic, catastrophe. Catastrophes break things apart often. So catabolic reactions break bonds. So a catabolic reaction also is going to pair with that exergonic reaction graph, right? Because we're breaking bonds, releasing energy. Anabolic reactions, here we have like these two distinct substrates coming in that are unbonded. They combine with the enzyme, and then we have a bonded product coming out. So we're building bonds in an anabolic reaction, so we're storing up energy in that bond, so that would be more likely to be an endergonic reaction. An example of a catabolic reaction that we've talked about before would be a hydrolysis reaction, right? And an example of an anabolic reaction would be the condensation reaction. So enzymes are going to be very specific to the reaction that they catalyze. They have very specific reactants. They are often called like a lock and key, right, analogy. So like the enzyme would be the lock, and then the reactant is called the key. So you're going to have a really specific enzyme for one kind of reactant. Right? So not all enzymes are going to be able to the enzymes aren't going to be able to be used for different types of reactions. Another word for reactants is substrates. So reactants, substrates, the substrates are going to bond with the specific enzyme. So if we have an enzyme catalyzed reaction occurring, the substrates are going to bond to the enzyme at something called the active site. So the active site is a spot on the enzyme that is shaped very specifically for its substrate, right? That lock and key analogy. Right, so we can see here's the enzyme sucrase, right? And that enzyme sucrase is designed to bond the substrate sucrose. So we can see this active site is really specific, right, for that shape of the sucrose molecule. One thing to note too is that enzymes that always end in ACE, ASE, right, and they're usually named for the thing that they are bonding. So sucrase is the enzyme for sucrose. Lipase would be the enzyme for like a lipid, and so they're very specific. So the enzyme and the substrate, right, are bonding together, and they create this enzyme-substrate complex, and that substrate, again, is bonding at the active site. So here we have the enzyme-substrate complex, where the substrate has bound to the active site. The reaction occurs, so here's the reaction occurring, and then we end up with some products. So in this case, our products are glucose and fructose. If you're breaking things apart, you could end up with multiple products, right? If you're building something, you could potentially end up with one product. Now that product gets released from the enzyme, and the enzyme can be reused to bond another sucrose molecule. So this enzyme could go back, and then we can see this is a cycle. It gets reused, and it could bond another sucrose. All right, so how do enzymes actually work? Like, how do they actually break these bonds or form new bonds? And there's some chemical interactions that can occur, or some mechanisms that it can occur in a catalyzing reaction. So one option is something called inducing strain. And this is where the enzyme actually like pulls or pushes on that substrate. So we can see this happening right here. It pulls or pushes on that substrate, and it puts it in this unstable transition state by stretching the bonds. And that's called inducing strain. It's pushing on it, stretching those bonds. It's in an unstable state, so then those bonds are going to break apart. Sometimes the enzyme just works by allowing for a place where the substrates can be in the correct orientation. So if the substrates are just like moving around in space, right, they're going to rarely bump into each other in the proper orientation to react at that collision. So the enzyme is going to lower the activation energy by bringing them together in the proper orientation. So they're going to bond to that active site in exactly the orientation that they need to be able to bond. Another option for enzymes working is just to add on chemical groups. They can take side chains from enzyme amino acids involved in the reaction. They can transfer hydrogen ions to induce the breaking of the bond. So they can actually add some chemical groups to either create a bond or break a bond. Many enzymes actually have some non-protein helpers that can play some roles in helping with some chemical processes like electron transfers. So some of these adjuncts are called cofactors. Some of the cofactors are inorganic, like iron, copper, zinc molecules, so they can help bind to the active site and participate in different reactions. And then some of the helpers can be coenzymes. So the cofactor is an organic molecule, it's going to be a coenzyme. Oftentimes the vitamins that you have to eat, we know certain vitamins are important, and those vitamins are important because they're acting as coenzymes. They're helping your enzymes with various reactions. Prosthetic groups are permanently bound to enzymes, and they're going to be non-amino acid atoms or molecular groupings that can do things like transfer electrons, bind electrons, convert light energy, some other things like that. Some of these molecules are going to increase enzyme activity, so they're going to be activators, and then some of these molecules are going to decrease enzyme activity, and they're going to be inhibitors. One of the things that we're going to be looking at more specifically, and we're going to do a lab around this, is reaction rate. And we can see from this graph, right, something should seem relatively obvious based on the activation energy stuff that we've already talked about, is that the reaction rate is going to be increased with the enzyme. So we can see that increased reaction rate here with the enzyme relative to the reaction rate here without the enzyme. We can also see that the reaction rate is going to increase with increased amount of substrate. So as the concentration of the substrate increases, essentially when there's more stuff to react with, the reaction is going to be happening faster, which makes sense intuitively. However, it gets interesting because we can see that the reaction with the enzyme is increasing really fast here. So right at low substrates, right, there's going to be lots and lots of reactant or substrate relative to the amount of enzyme. So lots of options for that substrate to bond to. But as we start increasing substrate, that reaction is going to level off at some point. So we can see it's leveling off here at this maximum rate. So this is the maximum rate, the maximum speed that the enzymes can start reacting with substrate to produce product. Now, why that maximum rate is leveling off here, even though substrate is still increasing in amount, is because we have a limited amount of enzyme, right? So our reaction rate is going to be limited by how much enzyme we have. Because at some point, all the available enzyme molecules are going to be bound to substrate molecules. So the active sites are saturated. So the enzymes are working as fast as they can, and they're all bound to substrate, and they can't work any faster. So any additional substrate just doesn't have anywhere to bond until that reaction occurs. If we were to increase the amount of enzyme overall, then our maximum rate should increase with a little bit more with the concentration of substrate. There are a few other things that can influence how fast a reaction is occurring, and those are inhibitors and activators. So there's a couple different categories of inhibitors. There is irreversible inhibition. So this is where an inhibitor covalently binds, so strongly binds at an active site, and the enzyme gets permanently inactivated. This is not going to be very common in cells because enzymes permanently inactivated can't be recycled, and that just seems kind of wasteful. So this is less common in cells. More common is to have reversible inhibition. So where a molecule is going to bind to the enzyme, it's going to inhibit that enzyme for a period of time, and then that could be reversible where the enzyme opens back up again and is able to bind substrate. There are two types of reversible inhibition, competitive and non-competitive. We can see down with this graph, if the purple line is our normal enzyme catalyzed reaction, the green line competitive and the blue line non-competitive inhibition are both going to be decreasing the rate of reaction. Competitive inhibition, we can think about this as the competitive inhibitor is competing with the substrate for the active site. So it's trying to get into the active site and bind to that active site. If the inhibitor gets into the active site, right, it's going to physically block it. We can see that right here. Right? It's going to physically block it so that the substrate is not able to bind. Right? Since they're both trying to squeeze into that active site, competitive inhibition is going to reduce the reaction rate, but not quite as much as non-competitive inhibition, which is a little bit harder to reverse. So non-competitive inhibition is where we have an inhibitor right here, right, binding somewhere else on the enzyme. We'll talk about this in a minute, but it's called the allosteric site. So a non-competitive inhibitor binds somewhere else on the enzyme, and what that does is it changes the shape of the active site so that the substrate can't bind or the reaction can't occur. So since that non-competitive inhibitor would have to then be removed and the enzyme would have to reset, this is going to be harder to reverse, so it's going to reduce the rate of reaction even more than a competitive inhibitor would. So competitive inhibitor is competing directly for the active site, is blocking the active site physically so that substrate can't bind. A non-competitive inhibitor binds somewhere else on the enzyme and then changes the active site so that the substrate can't bind or the reaction can't happen. Non-competitive inhibition can also be called allosteric regulation because the site where the inhibitor binds is called the allosteric site. So we can see here's our inhibitor molecule, our non-competitive inhibitor. It's binding at this allosteric site, which is not the active site. When it binds, we can see that active site here is changing shape, so it's shutting down versus being open right here. So it's shutting down and changing shape so that this substrate is not able to bind. We can also have allosteric activation, so we can actually start off with the enzyme closed, right? So here we can see the enzyme is starting closed, right? And then it's going to require an activator molecule to open it up. So the activator has to bind again to that allosteric site in order for the substrate to be able to bind. This might be a case where maybe you only want the substrate to be open when another type of molecule is present. We can tie this allosteric inhibition and activation into thinking about feedback inhibition. So this is where you get like a kind of train of enzymes all working in one big pathway, right? So here we've got enzyme 1, right? Enzyme 1 is bonding this green triangle substrate, and that's producing this product, this purple product. The purple product is then going to be the substrate for enzyme 2, right? So that purple product turns into the substrate for enzyme 2. Enzyme 2 creates this product B, a blue product, right? And then that blue product is going to become the substrate for enzyme 3, right? Enzyme 3 turns that blue substrate into this yellow end product, and then the yellow end product can actually go all the way back, right, and inhibit that first reaction that created. It can act as an allosteric inhibitor and inhibit that reaction of the substrate that kicked off the whole pathway. You might want to use feedback inhibition where you want something to accumulate, and then once that thing starts accumulating, it can shut down the whole pathway so that you don't make extra and like waste chemical resources. So maybe this end product needed to build up a little bit, and once it builds up a little bit, it's able to go back to the front of the pathway, slow it down, right? Because remember that it's not going to shut it down completely, but it's going to slow down this pathway so that we're not wastefully producing products that the cell doesn't need as much of. Something that we're going to be thinking about in our lab is the influence of environmental conditions on enzymes. Enzymes often have really specific conditions that they can operate best in, and pH and temperature are going to be two of those. And we have these really specific conditions because the enzyme could have some chemical groups that react best or are folding best at certain pHs, or increased temperatures are going to change the actual enzyme shape. So we can see we have 3 different enzymes here, and they all have these optimal pHs where the enzyme activity, the reaction rate, is at its highest point. Now, pepsin has a really low reaction rate, but pepsin is an important enzyme in the stomach. So it needs to be able to tolerate and work best at that really acidic environment. In contrast, chymotrypsin is in the small intestine, which is less acidic, so it needs to operate to help to absorb food items and nutrients at a higher pH. Similarly with temperature, right, enzymes are going to have an optimal temperature, and that optimal temperature is going to depend on the reaction that they are catalyzing and the environment that they are living in. Right? So here we see this optimal temperature is 40 degrees Celsius. So as we approach that optimal temperature, the reaction rate is going to increase. Right? It increases because increased temperature helps the rate of reaction. But then once we get beyond that optimal temperature, the reaction rate is going to drop off. Right? This could be because increased temperatures beyond this optimal are actually going to change the structure of the enzyme, kind of allowing that enzyme to unfold, right? When we talked about enzyme and protein folding, it's going to allow that enzyme to unfold, which is called denaturing. And if that enzyme starts to come apart and denature, it changes the shape of the active site, which makes it less effective. And again, temperature is going to be really specific to the kind of reaction and the enzyme and where it is. So like a bacteria in a hot spring is going to have much higher temperature, optimal temperature ranges for its enzymes than we might in our body. All right, that's it for enzymes for now. We're going to use all this information, we're going to answer questions on it, and then we're going to get ready to do a lab on optimal environmental conditions for enzymes.